which leads us to the question, what are the ecological drivers of brain size evolution? A recent work has begun to address this in captive populations of guppies. So artificially selected lines of guppies for large and small brains were used to look at the role of predation in the evolution of this trait. These guppy selection experiments have shown that brain size evolves rapidly in response to directional selection. And these results also indicated that as brain size increases, we see increases in anti-predator behavior as well as increases in overall cognition, and which ultimately results in decreases in susceptibility to predation. And so while these artificial selection experiments provide excellent support for the role of predation in brain size evolution, they cannot account for ecologically driven mortality or other ecological interactions in the natural community. And so we set out to determine if predators select for larger brains in natural populations as well. And so our natural populations of killifish are located on the island of Trinidad and right in the northern mountain range. So we were on the southeastern slope. These fish were collected there in streams that have now been studied for over 4 decades. And while the favorite study organism on the island seems to be guppies, we actually looked at Trinidadian killifish, Rivulus hartii. And these guys make an excellent study organism. They're egg layers, they're external fertilizers, easily transported, lab friendly. But one of their most important characteristics are their large dispersal capabilities. And this large dispersal capability is the reason why these fish are observed across stream communities throughout the island. In our study system, our streams have 3 different communities which differ in their predator assemblages. And each of these communities are separated by these large waterfalls, and these physical barriers prevent the movement of predators between these sites. So in our lowland rivers, in our high predation sites, we have large placivorous predators that feed upon Rivulus, again truncated by this waterfall. And then we have Rivulus guppy sites where Rivulus and guppies co-occur, again separated by waterfall. And then in the headwater tributaries, we have Rivulus only sites. And what makes this a really excellent system to study is that these sites are only tens of meters from each other, and they also do not differ significantly in their habitat characteristics or their water quality parameters. So this makes it really great to look at the role of predation in a controlled natural setting. And so the interactions between predators and Rivulus have been quantified. Specifically, we have our Rivulus only sites, which of course don't have predators, but our Rivulus guppy sites and our high predation sites obviously do. And these sites differ in their rates of predator-driven mortality. So in our high predation sites, we have these large fish that like to feed upon juvenile Rivulus as well as adult Rivulus. And then in our Rivulus guppy sites, we have guppies who like to feed upon the juvenile Rivulus. And this is largely because of size constraints. And so it's not surprising that our Rivulus experience higher rates of predator-induced mortality in the high predation sites when compared to the Rivulus guppy sites. And work completed by Walsh and Resnick looked at how life history evolution traits differ in result to this predator-driven work, these differences in predator-driven mortality. So fish were collected from Rivulus only sites, Rivulus guppy sites, and high predation sites, and were used in a common garden experiment to look at the genetic basis of the differences that are observed in their life history traits. And what's important to note, I guess, is that Rivulus only and high predation sites act as extremes in these trait values, and then Rivulus guppy values are usually found somewhere in the middle. So, for example, age at maturity for Rivulus from Rivulus only sites is much later in life, whereas Rivulus from high predation sites experience sexual maturity earlier in life, and then Rivulus from Rivulus guppy sites are found somewhere in between. And the fish that were actually used in this research were used for our experiments. So these fish are incredibly well traveled. They were collected in Trinidad back in 2005, had a quick layover in Houston, and then were brought back to UCR for Matt's PhD work, where they were the parents for the Common Garden experiment. And the offspring from the Common Garden experiment were preserved, traveled to Yale with Matt for his postdoc, and then came back down to Texas in 2012 when he got his faculty position at UTA. And then this past fall, they were dissected for their brains in our lab. And so what we know from the Guppy selection experiments would make us think that the Rivulus from the high predation environment should have a larger brain size when compared to the Rivulus from the Rivulus-only sites. And we actually found the opposite of this. So males from the high predation localities have smaller brains than those from the Rivulus-only sites. However, we did not observe the same in our females. So female brain size does not differ significantly between our high predation and Rivulus-only locations. And so our predators are selecting for smaller brains. We also assessed our fish for evidence of the expensive tissue hypothesis. So we had dissected their guts and weighed these as well. And we found no evidence for the expensive tissue hypothesis. So correlations between brain size versus gut size were non-significant. And then we also looked at potential trade-offs between reproductive investment and brain size. And again, these trait correlations indicated that there was no evidence for trade-offs between reproductive investment and brain size. And so now I've talked about how the Rivulus brains from high predation and Rivulus-only sites differ, but I've left out the Rivulus-Guppy relationship. So we actually found that brain size does not differ between Rivulus-Guppy locations and Rivulus-only sites, indicating that predation early in life does not select for differences in relative brain size. And so moving forward, this leaves us with two pretty big questions. Why are small brains selected for in risky environments, and why are larger brains favored in the absence of predators? So to address the first question, we know that in high predation locations, fish or animals in general usually will engage in riskier behavior, and that some of these risky behaviors are associated with increases in fitness. Rivulus are known to be bolder and to move around more in high predation locations when compared to Rivulus-only sites. And so it's possible that this could be the explanation as to why small brains are being selected for. And as for larger brains being favored in the absence of predators, other work on fish has shown that fish that aren't exposed to predation experience faster rates of learning and are also have higher levels of overall cognition. And so it's possible that our Rivulus in these Rivulus-only locations, which engage in more foraging behavior and aggressive interactions with males, might be experiencing something like that, in which it's more beneficial for them to have these larger brains, which is associated with increases in spatial learning abilities. So what we're thinking is that these differences in brain size underlie these behavioral differences observed in Rivulus. And more work is needed to be done with that as we're moving forward. And with that, I would like to thank my advisor, Matt Walsh, David Bresnick, and the grant that he received from NSF